Stephen, faculty members of various departments, delegates who have come from other institutions, dear students. For me, it is a sort of a homecoming after about 37 years precisely. When I was here during 65, 67 period, still that is the classroom. The ground floor was the classroom for the first group, A2 batch. And for the first year, that is during 65, 66, our classroom was on the first floor, A1. And this building was not there. I think it came quite later. And also the chapel. In fact, that chapel was one of those, that old traditional type tiled building. Now I could see a remarkable change there. A wonderful, very beautiful chapel has come up of late. And I have still vivid memories of those days. I was deeply influenced by the then principal, late Father Theobald. And also a couple of teachers, particularly, although I changed over or I continue with the science, a teacher who influenced me really remarkably was the then teacher in the English department, late Sister Amanda. She was, I had a sort of a very intimate, personal relation. She was so affectionate to me. Then another teacher who influenced me remarkably was the late Matthew. He was then the head of the Department of Chemistry. I still remember an incident because although I think the classes were supposed to start on 14th June 1965. I still remember that. But I could not come to the class during the first week. I was down with hepatitis. So I came after about 10 days to the class. And then, I don't know what is the position now, then the system was, you have to sit according to your register number. And the register numbers are allotted alphabetically. Though I was maybe 4, 5 or 4, 6 tall, too small. And particularly in the school, I used to sit in the front bench. What happened was that my number was 1062. So I was in the middle row, 7th row, in the 7th bench in the middle row. So I came to the class. The first hour was of course English, but in the second hour was chemistry. I was very unsettled. Immediately he asked me, stand up. I stood up. He asked, what is the symbol for silver? Absolutely, I had no idea. Then just out of common sense, I said SI. Immediately said, stupid, how can you say this? So I started crying. Because I was totally, I came to the class for the first day. You remember the transition from a school to the college. All sorts of problems were there. I just could not resist. Then, after the class, somehow it seems he realized that something went wrong. Then he called me to his room. That complicated further. So I don't know what is going to happen. Whether he is going to suspend me. When I went there, he was so sober. He asked me to sit down. See, remember, does any teacher tell you the student to sit down? He asked me to sit down. He asked me what is the problem. So I told him that this is the first day for me in this college. I have absolutely no idea. Oh, then he said, immediately he said, okay, doesn't matter. But then from that day onwards, he was so considerate. And in fact, the most attractive subject for me then was chemistry since then. I continued for my BSc in special chemistry. Then, of course, I switched over to biochemistry, and then that interface, although I started from through the mathematics stream and now moved on to the interface and now with cell biology and molecular biology area. So I still have vivid memories of those days, although it's some 37 years ago. Deeply impressed, and now I could see the wonderful buildings, the progress of this institution over the past several years. At that time there was only the pre-degree, but when I left the following year, this college was upgraded. Then they started BSc and BA courses. I'm deeply impressed by the present status of this institution and the people, particularly the faculty members and good students who are behind all the activities of this institution. In fact, Stephen, as you could see, is so persistent and perseverant that unless you agree and come over for a function, until and unless that is materialized, he won't leave you. And in this case also, the situation was not at all different. He saw to it that I come here and participate in this function. I am personally thankful to him for all these arrangements he has made. And the topic which I am going to discuss, the title of the talk, as you could see from the, says Book of Life. I think it falls very much within the theme of the seminar, Genomics, Proteomics and Society. And in between I will arrange to bring in the issues which are in the area of genetics, which are relevant to the society, the societal development, and the implications of these new developments to the society. So the topic which I would like to discuss briefly, I would try to restrict about 45 to 50 minutes time. Sometimes it may spill over a bit depending on how we go on. The topic, of course, is figuratively written as Book of Life. As you know, the basic principles in heredity involve the existence of a genetic material which can safely replicate, so that whatever information is there in the genetic material will be faithfully transmitted to the daughter cells and to the offsprings. And the classical experiments carried out in 1940s conclusively demonstrated that the chemical that is responsible for carrying this genetic information is not protein, not carbohydrate, it is nucleic acid. The classical experiment of transformation of bacteria, the non-virulent pneumococcal bacteria could be transformed into virulent bacteria, that is the pathogenic bacteria, by treating these bacteria with the pure DNA isolated from virulent bacteria. That is the classical experiment of Griffiths, which conclusively demonstrated that DNA is the genetic material. Then the question was, if DNA is the genetic material, how this genetic material can carry the information, how this information can be transmitted to the daughter cells. The material should have the characteristic property so that it replicates, and as the new cells are formed, this replica, identical replica will be there. And in this direction, the first All living organisms in this plant, right from bacteria to highly complex organisms, are made up of the genetic material deoxyribonucleic acid. Then the question is how this deoxyribonucleic acid can serve as the genetic material. As you know, the paper which appeared in Nature in April 1953 by James Watson and Francis Crick, they announced they proposed a new structure for the DNA, the double helical structure for DNA. It was a small paper, 958 words, paper which appeared in Nature. Last year we celebrated, people all over the world celebrated the 50 years of this publication. If you could see the last sentence, a very dormant type of sentence, it has not escaped our notice that the specific pairings we have postulated immediately suggest a possible copying mechanism for the genetic material. That is, this parent material can be copied to form new material which are identical to the parent cell. What are the essential features of this double helical structure proposed by Watson and Crick? They proposed that DNA is a double helix consisting of two polynucleotide strands. These two strands have a remarkable property that the bases in these two strands are complementary to one another. That means if one of the bases in this strand is adenine, the corresponding base in the complementary strand is thymine. And if the corresponding base in this strand is guanine, the corresponding base in the complementary strand is cytosine. That immediately leads to the conclusion that if these two strands serve as template for the formation of new strands, as the new strand formed will be complementary to this one, and when this is serving as a template, the new strand formed will be complementary to that one. With the result that you get two new DNAs, the sequence of these two new DNAs will be complementary to, will be identical. So if this is the genetic material, and if this genetic material carries the information in the sequence of the bases, then this provides the right mechanism to transmit this information as the DNA replicates to the daughter cells. And this was the remarkable paper for which, in fact, this led to the development of the major theme in the biology, which led to the new branch of biology, molecular biology, and the central theme which was proposed at that time was called the central dogma. That is, the DNA has a self-replicating mechanism. This being the genetic material can replicate to produce identical DNA. So whatever information is there in the parent genetic material will be there in the daughter cell. Second, to carry out the functions in the cell, this has to be transcribed into another form called the ribonucleic acid, RNA, and this RNA is then translated to protein, and the proteins, large number of proteins are present in the cells. These proteins are the action molecules present in the cell which carries out a large number of functions, structural functions, biochemical functions, metabolic functions, recognition. Large number of functions are mediated through these action molecules called the proteins. And multiple cells associate to form tissues and the various organisms. So this is the classical central dogma wherein the basic information resides in the DNA, which is then transcribed and translated. Now how is it possible to look at this information? So they have established that this is the DNA is the genetic material. So DNA has the information must reside within the bases. So how is it possible to transcribe this information? What is the biological mechanism? I take an analog situation, an information transfer machinery what we are using, namely the language. We convey our ideas through a particular language. Number of different languages are there. English language, Malayalam language, number of different languages. But basically,
You know that all these languages have a characteristic alphabet, a characteristic lexicon. English language, for instance, if you take, there are 26 alphabets, there are about 35 characters, 26 alphabets, 89 punctuation marks. Similarly, other European languages have almost similar number of characters. Similarly, the Oriental languages also have well-defined alphabets, specific scripts, and specific rules for producing sentences, conveying the ideas. And you could see in English words of variable character length. Say, for instance, I, of 1 letter, 2 letter, 3 letter, 4 letter, 5 letters, and even see, look at the longest word, anti-disestablishmentarianism. So many words. So the possibility is that you can have a word with 1 letter or even with 15 or 20 letters. It varies from language to language. And there are also rules to start a sentence with a capital letter in English, then to stop the sentence with a full stop. There are phrases, clauses, and a number of sentences can constitute a paragraph, number of paragraphs can constitute a chapter in a book, and number of chapters can constitute a book. And even in large books you could have a number of volumes of books. So that is possible. Now look at the genetic language. That is what we are looking at. The genetic language is very simple. There are only 4 letters. Unlike the English language, where you have 26 letters, in the genetic language we have only 4 letters, 4 alphabets, and these are the 4 bases: adenine, guanine, cytosine, and thymine. And unlike in other languages where you can have a word with 1 letter, or a word with 2 letters, or a word with 3 letters like that, here we have only 3-letter words. We have only 3-letter words. That is, of these 4 letters, adenine, guanine, cytosine, and thymine, each word is made up of 3 letters. That means it is a very small lexicon. It's a very small lexicon. Out of these 4 alphabets, 4 letters, consisting of 3-letter words, the maximum that we can have is 64 words. That is the maximum that we can have. Just 64 words. See, look at that. It's a very small lexicon. It's a very small lexicon, whereas in other languages, in English using 26 letters, you can have millions of words. If you look at the Chambers English Dictionary, you could see every time they revise, the size also increases. The number of words, new words come, new words are coined. But here it is defined. It's a very small lexicon, just 64 words. And these words are called the codons. No more than 64 words. There are certain words which are specifically intended for starting the sentence, like you start a sentence in English language with a capital letter. There are certain words, here it is a 3-letter word, where it's called a start codon. And similarly for full stop, you can have stopping words or called stop codons. But the most important thing is that unlike other languages, unlike the languages which we use for conveying the message, this genetic language, no matter whether it is a bacteria, whether it is a virus, whether it is a plant, whether it is a highly complex mammalian organism, including the highly arrogant human being that we are, it is highly universal. For words are the same for all the organisms. For the genetic language is written a set of words which is universal for all the organisms. And this can therefore be referred to as the book which is written, the genetic book which is written using these 64 words lexicon, or can be referred to as the book of life. And the genetic instructional language which is written in these 64, in fact 61 words, codon, and the stopping codons. So using that, we have to transcribe or translate this genetic language into some other language, say Hindi to Malayalam, so that a person who knows Malayalam can understand Hindi, or from Malayalam to Hindi, like that. So it should be translated to a different language. And what is the language to which it has to be translated in the cells? The action molecules, the proteins. So this has to be translated into the 21-letter action molecule, the language of proteins in the various cells. And the sentences made out of the 64-word vocabulary, we can consider the concept of gene in a different perspective. Say each gene can be considered as a sentence or even as a small story written in a small paragraph. And the morphological unit in the cell, namely the chromosome, each chromosome can be considered as a chapter of a book. So each genome can be considered as a genetic book comprising of a collection of chromosomes. And you can see that genomes or book of varying chapters. For example, E. coli, the smallest bacteria, which has one chromosome, or the book of life for the genetic book for E. coli can be considered as a book of single chapter. A fruit fly, the symbol Drosophila, which is used extensively by developmental biologists to study of these days, to study developmental biology. It has a genetic book consisting of 4 chapters. Yeast, it has 17 chromosomes. Dog, 37 chromosomes. The hermit crab, 127 chromosomes, 127 chapters. And the humans have 23 pairs of chromosomes, or our book of life consists of 23 chapters. So the human genome made up of 23 chromosomes and roughly about 4,000 genes, and the total 40,000 genes, and the total length of the DNA in this genome is about 3.2 gigabases long. That means that is about 3,100 million bases. In American language, you call it 3.2 billion bases. For us, it is 3,100 million bases. In the size of that, the book of life for us. And the genes carry the characteristic genotype, which is translated as typical traits into the phenotype, which is expressed through the proteins. This is the morphological analysis of the chromosome. Is in fact numbered in decreasing size. The one which is the largest one is referred to as the chromosome number 1, and the one which is the smallest size is referred to as the chromosome 22. There are 2 pairs of the chromosome. Each pair is inherited from the parents, one pair from the father, second pair from the mother. And there are also, you could see the remarkable difference in this 23rd chromosome. These are allelic forms, whereas here these 2 are 2 different chromosomes. One is the X chromosome and the other one is the Y chromosome. In male you could see XY chromosome, whereas in female you have XX chromosome. See, look at the difference. Just this is the difference, and you see the remarkable difference in the society not only in terms of the gender issues, so many issues out of this poor chromosome here. If you come back to a single chromosome, you could see that the chromosomes, they contain the bases, the deoxyribonucleic acid bases, and the genes are located here. It can be gene 1, gene 2, gene 3, etc. And this DNA is present essentially in the nucleus. A little bit of DNA is present in other subcellular organelles like mitochondria, but the major genetic material is present in the nucleus. So if you look at the dimensions, the size of this, say if you take say the distance between a bases from the double helical structure, the dimensions of the double helical structure proposed by Watson and Crick in their classical paper, the length of the 23 pairs of the chromosomes in our DNA would come to about the time, the distance covered by light from sun to earth 7 times. See, imagine the length of the molecule that could be. Or if you want to read the bases, say the adenine, thymine, guanine, cytosine, and you take 1 second each, you take 1 second each to read base by base, adenine, thymine, guanine, cytosine, then the time taken will be of the order of 10 to the power of 200 years. See, that is the size. But still, this material is present in this cell, and that too within the cell in the nucleus. That means such a huge structure must be tightly condensed. Such a condensed material is packed within the nucleus, and the condensation takes place by combination with the proteins, and you know that for biologists you know the nucleosome structure through which the nucleic acid in combination with the protein is packed and kept in the nucleus. The transcription of this DNA replication takes place in the nucleus, transcription to RNA takes place in the nucleus, the RNA is then translated into proteins, and the proteins then carry out the various cellular functions. Now a major project, I come to some of the recent developments in this area. A major project was undertaken recently was to read the human genome. It was referred to as the Human Genome Project. By reading the Human Genome Project means to read the arrangement of the bases, the sequence, the bases of the DNA of all the chromosomes, identify the genes, then compare these genes with other genes of other organisms. So to read that, we must know where it starts, where it stops. We should be able to read it correctly. I just give you one example if we do not know how to read it correctly. Say, look at don't read the bottom line, read the first sentence. He says Crick is stupid. Very atrocious statement if it is so. He says Crick is stupid. But the next sentence, you put a comma there. He says Crick is stupid. That means Crick is saying that. But actually, if you read it correctly, you can see that he who says Crick is stupid is stupid. See, look at how the message, that meaning changes. So you should be able to read it correctly. Incidentally, as you know, on 28th of last month, Francis Crick died at the age of 88. He was suffering from colon cancer. He was the person, the architect, along with James Watson. He was the architect of the double helix. He died on 28th of July. For quite some time, he was suffering from colon cancer. He was not even available for his comments when the Human Genome Project was announced. In fact, I would dedicate this lecture to the memory of Sir Francis Crick. So we should know that, we should know to read it correctly. If you do not read the differently, then the message that goes out will be different. Same holds good for the reading of the genetic book. So they read it letter by letter, put it into codons, translate into proteins, and see if this protein is present. Just sequencing doesn't mean that it is expressed. So see whether this protein is present. So this is going to be the ultimate thing. The reading the code words, DNA is a double-stranded, as you know, it's a double-stranded molecule which has two strands, a sense strand, matching strand, and if this is one of the strands, then the corresponding strand will be complementary to that. For a position T, there will be another base at the complementary strand will be adenine, adenine thymine, cytosine corresponding guanine, adenine thymine, etc. This is the sequence of the complementary strands. And when this is the arrangement of the bases, adenine in one of the strands pairs with the thymine in the opposite strand through hydrogen bonds. Adenine, the stability of the helix is maintained through the hydrogen bonding interaction between adenine and thymine and guanine and cytosine in these double helical strands. And the sense strand, for the transcription process, the sense strand is transcribed to a message. The only difference is that instead of thymine in the messages, you have uracil, whereas the adenine, guanine and cytosine are the same. So this is the message that is formed, and this message which is formed by the transcription, that is you are transcribing one language into another form, and this is then translated into the protein language, and you get, these are the codons AUG, UUA, AGG, etc. This is translated into a protein sequence using the 3-letter code, and this is the one which is the starting codon, and these are the ones which code for specific amino acid. UUU code for amino acid phenylalanine, AGG for arginine, UCA for serine, etc. And UAG is the stop codon. It does not proceed further, it stops there. So this is the protein that is formed during the translation process of the messenger RNA that is formed by transcription of the gene. Now the Human Genome Project was proposed to sequence the entire bases in the 3.2 gigabase, that is 3,100 million bases in the human genome. That was the whole idea. But the project initially was could not take off for several reasons. It was controversial from the very beginning for the simple reason that people raised several objections. Major objections included that this is going to be a major project in biology. So in other areas of other branches of science, say you remember the project for development of nuclear bomb. You remember the Apollo mission for moon landing. Number of such major mega projects were successfully executed in other branches of science, but in biology it has never been done. So the scientists working in other areas of biology feared that if such a major project is taken up, then they will be starved of funds for their research. So there were tremendous resistance from biologists themselves to start with this project. But the important problem was that the technologies that are available were not sophisticated enough, not fast enough, not sound enough to take up such a major project for sequencing 3,100 million bases. That was the problem, and also the resources required in terms of finance, in terms of the technical resources, it was not sufficient. But there were again different groups who supported the launching of the project. At the same time, there were different groups who opposed that. Two major agencies which came forward to start this project was, one was the Department of Energy. See, that has nothing to do with normally, that has nothing to do with the biological, the sequences of the gene. The Department of Energy of the US government pledged about $12 million to start this project. And about $18 million were pledged by the National Institute of Health, USA, to start this project. A committee with Dr. Bruce of the University of California at San Francisco was constituted to work out the clear proposal to start this project. And officially in 1988, the committee announced the starting of the project. And according to that, James Watson, none other than James Watson, was asked to be the chief executive officer of this new project which was started in NIH. And in 1988, they announced that officially the Human Genome Project will start on the 1st of January 1990. As the US Human Genome Project is initially proposed as a 13-year project. The project originally was planned to last 15 years, provided sufficient technological and financial resources are available. And the original estimate was 15 years duration, $200 million per year. And the project goals were to identify all the genes in human DNA, determine the sequence of nearly 3.2 billion base pairs in the DNA, store this information in databases, improve the tools for the data analysis because it's a huge amount of data. Simple manual analysis is not sufficient. So sufficient technological tools, particularly computational tools, must be developed to analyze this data and see whether useful technologies can be generated from this, and if so, it has to be exploited properly. And along with that, there were a number of issues to be addressed, particularly looking at the resistance for the recombinant DNA technology in 1970s, because there was stiff resistance against recombinant DNA technology in 70s. And in fact, it dampened the speed with which the recombinant DNA technology could be developed. Originally, this was started in 1972, but it took another 7 years to have insulin produced through this recombinant DNA technology, mainly because of the apprehensions expressed not only by the activists, even by biologists themselves had expressed the serious apprehensions. So it took quite a bit of time before the fruits of this new technology could be realized. So they made it a point that along with this, the issues, particularly the ethical, legal and social issues that may arise from this project must be addressed. Now, as you know, the important milestones was in June 2000, precisely on 26th June 2000, the classical press conference you must be remembering, the press conference held by Bill Clinton, Francis Collins and Craig Venter, the scientists who spearheaded the program, and Bill Clinton at White House. And at the same time, Tony Blair and John Sulston in London announced the unraveling of the Book of Life. The rough draft of the human genome was published, was announced, and the real data came in February 2001. The data generated by Craig Venter appeared in Science, and the data generated by other groups but led by NIH appeared in the journal Nature. This is just what has happened. I just go through some of the intricacies, some of the historical part incidents in the Human Genome Project. See, as I said earlier, the Human Genome Project was launched initially through a series of discussions by eminent biologists. James Watson was actively involved. He undertook the program. He agreed to head this program at NIH. And then at NIH, see this person, Craig Venter. Craig Venter, a Vietnam War veteran, he was in Vietnam with the American Army. He came back from Vietnam after the war. Then he thought that this is not a good area for him. He moved into academic field. He took his PhD. Then he joined NIH in the immunology division. He joined as a scientist in the National Institute of Health, Bethesda. And he was associated with sequencing, partly with this sequencing business. He believed in speed. he was not keen on waiting for data to be generated through systematic means. He thought that it would be better to go for rapid sequencing. And for that he undertook instead of going for all the sequences, he thought that it would be better to take the messages. Anyway, in each cell, the DNA is transcribed and you get the message, and from the message you get the protein. So why don't you go for the messenger RNA? Why don't you go for the messenger RNA and then look for the corresponding DNA? You go use the reverse transcriptase, and using the reverse transcriptase with this messenger RNA, we can take the cDNA and then sequence that cDNA. So he started doing this, similar to what Sidney Brenner in the Medical Research Council in England was doing. And he started, while doing this, he started patenting this. He started patenting this, for which he established a company called Tiger. And using whatever data comes from this, whatever genes he could patent, another company could take the patent rights, for which he gets the money. So the company which he plotted for this was called Tiger, the Institute for Genomic Research. In short, the Institute for Genomic Research. It is not, although it is TIGR, he used to pronounce it as Tiger, TIGR, because he wants to have that stamp of that. And this was associated with another company called the Human Genome Science. The Human Genome Science said that whatever patent you take, we take the patent, commercialization of the patent for 6 months. After 6 months, if you don't want that, you can sell it. And then came a pharmaceutical company anticipating tremendous potential for this new data. A pharmaceutical company called SmithKline Beecham came into the field. They invested money in this project, and these three group of people, the Tiger, HGS and SmithKline Beecham company, they started patenting this. And incidentally, it was a speculative patenting, because they didn't know what this gene was. They didn't know the importance of the sequence at all. But still they patented it. But at some point of time, it turned out that one gene which they patented, without knowing what exactly is the function, without knowing what exactly is the product, one gene which they patented happened to be a gene which is responsible for the progression of the HIV infection in the T cells. So that means it has a tremendous potential. This gene can be targeted for HIV treatment. Realizing the potential, another company, a competitor of SmithKline Beecham, Merck company, came forward and they started funding projects in other groups, namely the NIH and various laboratories which were sequencing the human genome. And one of their conditions was, the SmithKline Beecham condition was that you should patent it and the proprietary rights of the patent will remain with us. But what the competitor Merck wanted was that you should not patent it, you should publish it. So there, once you publish it, it becomes part of the public domain that will effectively prevent Tiger, HGS and SmithKline Beecham to patent that. But this sort of patenting the genes annoyed Watson. He resigned from that, and in 92 he left NIH program and he joined back the Cold Spring Harbor laboratory. Then came Francis Collins as the head of this program in NIH. But later on, it was a real race. Not only that, Craig Venter subsequently, they realized that, so you see that the sequencing is a laborious process, particularly because the size of the genome is sequencing methods available till then. Particularly the one which was available was the dideoxy method developed by Frederick Sanger. You know the dideoxy method. That was the method which was developed by Frederick Sanger. But that method which was developed by Frederick Sanger, the system involves 4 different types of dideoxynucleotides: dideoxythymine, dideoxyguanine, dideoxycytosine, and dideoxyadenine. Not only that, you have to use the 4 different reaction systems to have the sequence of a small segment. And then after using this reaction system, you separate this in a gel, and in a gel you have 4 lanes: dideoxythymine, dideoxyadenine, dideoxyguanine, dideoxycytosine. And this technique, technology-wise, although it was not that sound, but the technique by itself was really clever technique, because the scientific basis was quite good. But you cannot sequence by a single system not more than 100 or 150 bases at a time. So imagine the phenomenal size of the gene, the 3.2 billion bases. How long are you going to take to sequence this if you use this sort of a simple gene with which you can sequence maybe 100 or 200, at the most 1,000 bases? How long are you going to take that? So this was a compelling reason for going for the new technologies. So then came a new technology, that is, instead of using this sort of a dideoxynucleotide with radio-labeled tag, fluorescent-labeled compounds can be used. You can use fluorescent-labeled compounds. That is, you can use thymine, dideoxythymine with a particular fluorescent tag, dideoxyadenine with a particular fluorescent tag, with the result that for these 4 different types of tags, when you read under laser, these 4 bases, dideoxy bases, will appear in 4 different colors: a blue, yellow, green, and orange. With the result that you can read in the same lane. If you use an orange colored adenine, that will appear as orange when you use a laser beam to read that. And this was then developed, this technology was developed by a company called Applied Biology System, and they gave a prototype. Prototype means they developed a machine to test that machine, whether that machine is useful for sequencing. They gave a free of cost a prototype of this machine to this gentleman, Craig Venter. He made use of that, and he realized that this can be used for sequencing at up to about 100,000 bases. And then what they did was, this was originally developed by Hunga-Piller, Smith and Hunga-Piller. Smith and Hunga-Piller, they were working in Caltech, California Institute of Technology. They developed this machine. Once they developed this machine, they resigned from Caltech and they floated a company in association with the giant Perkin-Elmer company. And what they did was, they produced about 300 such machines. They produced a highly sophisticated automatic sequencing machine and gave these 300 machines to Craig Venter, who floated a company called Celera Genomics. He floated a company called Celera Genomics, and he gave this, Perkin-Elmer gave 300 machines at a very reasonable price. And they started in 1998 sequencing at a massive scale. Whereas, so this, in fact, it was a shock to other people who were following a systematic approach. Then they started pumping in money. They had to shell out a large amount of money to procure from the same company, Perkin-Elmer, another thousands of machines at a higher price. They also started using this automatic sequencing of the DNA. That is why I put the slide as the race, and this is the starting point. There are 2 ways of sequencing this. One is to go for sequencing in a very systematic, that is, you isolate each chromosome, that is, you have the 23 pairs of the chromosome, you separate each chromosome, identify the position of each gene in that. That is called a genetic map. And you do not know the sequence. Map is a rough indication where each gene is present. Something like you say that maybe Ernakulam is in between Trivandrum and Calicut. Certainly you know that this is between Trivandrum and Calicut International Highway. That is called a genetic map. That is, the gene 7 is in between gene 5 and gene 9. That much is that sort of a location. Then second thing is much more precise, a physical mapping. To be a physical mapping means what is the distance between Trivandrum and Ernakulam is, or Ernakulam is about 200 kilometers away from Trivandrum to the north, or Ernakulam is about how much, 150 kilometers towards the south of Calicut. This sort of a physical thing. That is, gene 7 is about so many bases away from gene 5 and about so many bases away from, in a particular direction, from gene 9. This sort of a map. You don't know what exactly is the sequence of the gene. You know that there is a particular gene and that particular gene is nearer to such and such a gene. That is all what you know. That sort of a rough map. That is the systematic approach. And then they decided that the next phase is the actual sequencing of this. So the first thing was to assign the genetic map and then assign each chromosome or segment of each chromosome to each group, and then they decided to go in a systematic manner. These are the two different ways of sequencing. That is, the first one, you take each chapter, namely each chromosome, fragment that in a very systematic manner, and for each segment you use the sequencing method and determine the sequence, and then you can put them together to deduce the sequence in a very systematic manner. Whereas this is called that is, you fragment this with the help of restriction enzymes, separate these fragments, sequence that, and then from the overlapping sequences you deduce the sequence of this one. Whereas the second one, this is called the public initiative. Public initiative, which was led by NIH under the leadership of Francis Collins, which was carried out in different laboratories in the US, France, Germany, Japan, and the United Kingdom. Whereas the other one, really reckless vendor style, Craig vendor style, what he did was, Malayaladil paranja vetti murichu. He just, the whole thing, he did not separate any chapter, any book. He just rashly cut the entire thing by a whole shotgun method. And whatever pieces you get, sequence that. It's a junk data, really junk data. And using this data, the clever thing was that the public initiative, they started work sometime in 91, and whenever they get the data, they put the data in the public domain. In the public domain it is available, it has been put into the databases. So it is already available. So what vendor did was, he started sequencing this randomly and then checked this sequence data with the data that is already available in the public domain. So he was cleverly using that data and then put this in a proper sequence. He started aligning that. So this is called the first one is a slow process, but as you can see, it is much more accurate. That is the original Human Genome Project's approach, HGP approach, or it is called the public effort, which was undertaken by various laboratories in the United States and other countries across the globe. Whereas the second one, they take the whole genome, shotgun it into pieces, sequence them, and using the computer algorithms and also the data from the public domain, you put them back in proper sequence. This is the private initiative of Craig Venter. This is faster but less accurate. That is why what you get is a rough data. The approach of Celera Genomics, the so-called private effort, was that. This is the point where till then the molecular biology biology converged with the, particularly the computer technology, which led to the emergence of the new discipline called informatics, for the data storage, for a retrieval of the data, and for the analysis of data. Now, of course, I would just look at, once we have read it, what do we do with it? So these two groups were going almost parallel to one another, and on June 26th, as in early May, Bill Clinton said, he said, fix them. That was his statement. You fix them. A remarkable statement, and this led to bringing together both these groups, and finally led to the announcement of the rough draft of this. By early May, both the groups started collecting all the data, but it was rather difficult to put this data, such an enormous amount of the data, put the data together. So what they did was, in one group, they hired about 300 computers, worked overnight, put the data together, and the deadline was fixed on June 25th. The public initiative could put together data, not entirely, it was a rough draft, put together the data on 22nd June, whereas the Celera Genomics lagged behind. He could put together the data only on 25th night. But anyway, on morning of 26th, both the groups came together and announced that. And the announcement was signified by the statement that this is the beginning of a new era. What do we do with it? What is this new era? What are the implications of this data? Along with this, while we were doing the sequence analysis of the human genome, a number of other genomes also could be sequenced, like E. coli, Helicobacter pylori, the cholera genome, Drosophila, Arabidopsis, yeast, even the simple worm C. elegans gene. So this data is already available. If you look at, I just go through briefly the important results or observations of this human genome project. There are only about 30,000 genes. Look at that. These 30,000 genes, this is just 1.5 times. This is just 1.5 times that of the simple worm C. elegans. C. elegans, it is an organism which is made up of about 920 cells. About 920 cells, and the genome size, the number of genes in C. elegans is not even half of an organism like us. We are always arrogant about ourselves, who have over trillion cells, 100 trillion cells are there. But still, this is the genome size. The number of genes, 1.5 times more than, and maybe twice that of the fruit fly, the simple fruit fly. The fruit fly has about 18,000 genes. Second thing is that these genes are distributed in the human genome randomly. There are areas where, called the urban regions, where you could see clustering of the genes, a high density of the genes, crowded genes in certain areas, whereas certain areas, these are called sophisticated, probably sophisticated urban areas, or desert regions or AT-rich regions where you don't see too many genes. But basically, what is then this 3.2 billion bases? If there are only 30 to 40,000 genes, what is the remaining DNA there? The remaining DNA there is referred to as the junk DNA. We'll come to that a bit later. Then there are certain protein coding domains in certain genes which are rearranged. That is, you could see different proteins with segments or domains which are identical to one another. And this occurs through gene shuffling, and certain segments of the genes are shuffled with segments of certain genes, with the result that even though you have only about 30 to 40,000 genes, it is possible to have 100,000 or even more proteins. Then there is a large amount of non-coded regions in the human genome, which is referred to as the junk DNA. And that makes our genome so large, it swells up. And the genome size of our genome size is about 200 times that of yeast, but it is only 1/200th of the small organism amoeba. This is because of the large amount of the junk DNA that swells the chromosome, our chromosome. And this junk DNA is in fact a fossil record. The repeats in it can be dated and traced. I will just give you one example later when we go back to the tracing of these, using this junk DNA to trace the past. They have reshaped the genome by rearranging the genes and making new ones. The junk DNA repeat sequences, the junk DNA contains certain specific repeat sequences, smaller repeat sequences, larger repeat sequences. And these repeat sequences constitute nearly 50% of the junk DNA in humans, whereas in mustard, say for instance, 11% of the junk, the repeat sequences constitute 11% of the junk DNA, or in C. elegans worm, it's about 7%, and in Drosophila, such repeat sequences constitute about 3%. There are large repeat sequence, smaller repeat sequence, mini satellite DNAs or microsatellite repeat sequences are there. And in fact, if you look from these, we humans are junk collectors. However, the amount of DNA has come down. If you look at the evolutionary scale, you can see that the amount of this junk DNA has come down over the last several years. A large number of these, the human genome looks like the mouse genome. That is, if you look at the genes, most of the genes that mouse have, we also have. Probably about 300 genes. We have about 300 genes which this symbol innocuous, I would say innocuous as far as from the laboratory point of view is considered, not from the field point of view. From the laboratory point of view, it is an innocuous animal for us. We have only about 300 genes which we do not have. A large number of these common genes are common metabolic route ones. Some others are special, say for instance, for immunity related ones and for cell death related ones. There are a lot of pseudogenes, that is, the genes cannot be transcribed, the whole gene sequence is there, but these are not transcribed and translated because they do not have the characteristic, the promoter sequence for starting the translation process. And most of these genes, over 900 genes, some of them have come from bacteria. That means at some point of time, there has been recombination with the human genome and the bacterial genome, which has resulted in the transfer of some of these genes from the bacteria to the humans. They are disabled such that they cannot be transcribed because they do not have the promoter sequence for initiating the translation process. Of course, these are the vestiges of our evolutionary past where many duplications of genes and DNA structures occur. Usually they resulting in the malfunction of the genes, say for instance abnormal diseases like gene duplication, chromosome duplication, such as Down syndrome. This is the most important thing. The single nucleotide polymorphism, there are where genes of two people differ but by one letter. Say for instance, I and my brother, we have inherited the genome from the same parents, my father and my mother. But still, physically, behaviorally, you could see a tremendous difference between us. And if you look back to the sequences where the genetic information is stored, you could see that there could be differences in single nucleotides at different points. And this is referred to as the single nucleotide polymorphism. And this is important in the sense that the individual behavior in terms of not only at the macro level, even at the micro level, in terms of the response to the ability to utilize drug, ability to respond to drug for a treatment, all these depend on the single nucleotide polymorphism or SNPs. All humans share about, that is very important. We need not be, say, if you look at the chimpanzee, different species, you could see remarkable difference in the genome, whereas all humans share about 99.9% of the genes and vary in but 0.1% of the genome. indicating the common heritage. Human genetic variation arises through a shuffling of but a few set of genes. I will just take another 10 minutes to look at some of the anticipated benefits of this mega project. Of course, this will improve our knowledge about the genetic makeup, the gene data, the sequence of the gene, and that will lead to a paradigm shift in the biological research. Already the conventional approach has been changed. Now you go for genomic analysis, that is the total genome expression in an organism, in a cell. You go for proteomics approach, you go for looking for the different proteins that are present in a given cell. You go for the microbial genomics and identify those genes which are different from the human genome, so that that particular gene which is present in the pathogenic microbe can selective drug development. So the whole scenario changes with the generation of these data from the Human Genome Project. Disease detection and correction to therapy, that is, instead of looking at from the symptom and the conventional clinical approach, you go for a much more drastic molecular approach for disease detection. Say for instance, there is coming up a new area called the treatment strategy based on testing of the genes in two different things. Identify those genes which are defective in an individual, or identify those genes which are responsible for particular disease. Now we know that not only for genetic diseases, even for other diseases, including the infectious diseases, there is a genetic component. You develop an infectious disease because there is a defect in the immune system. Otherwise you develop a resistance to that infection. That means the immune system has to be targeted. There is a genetic problem associated with that immune function. So the approach now is to go at the genetic level to look for the disease pattern. So the gene testing would be one approach, or if you can even think of gene therapy. There is already reports coming up, gene replacement strategies, gene replacement therapy. And also you develop new strategy for using drugs based on the assessment of the ability of an individual to detoxify a drug. We'll come to that a bit later. That means the anticipated benefits include improved diagnosis of disease. That is, from the conventional clinical approach, we switch over to the approach of molecular medicine. That is, you go for improved diagnosis of disease by looking at the genetic profile. You go for earlier detection of a genetic predisposition to disease. So for instance, now the genes in which mutations have been exposed to development of two separate genes have been identified, BRCA1 and BRCA2, breast cancer genes. Specific mutations have been identified, and these mutations have been identified in persons who have developed breast cancer. And it appears that the incidence of these mutations, so people with these mutations have a higher risk to develop breast cancer. So this is an earlier detection. This is a look to go for through a DNA-based microarray technique. You see whether such a mutation is there for that particular gene using gene probes. Similarly, colon cancer, the mutation in a gene which leads to the development of colon cancer has been identified. So now we know that it is possible to predict, that is, an individual whether has a higher risk to develop colon cancer. If the mutation of those genes, a particular set of genes are there, then the chances of developing that of colon cancer is higher. And also rational drug design. Say in the modern medicine system, you prescribe a drug. They develop a drug, say for instance antibiotics, and it's acting against a particular bacterial strain, or you develop tetracycline against a broad spectrum antibiotic. And they have standardized this dosage. Whether it is for me or for somebody in the US or for somebody in Africa, the dosage is fixed. But the ability of an individual to utilize this drug will be different. There are different enzymes in these organisms which have to metabolize these drugs. So the ability of these organisms to utilize this drug is critical in determining the level of this drug in the body. For certain people, the metabolizing ability may be higher. For such people, you may have to give a higher dose so that the reasonable amount of the drug will remain in the circulation so that it will be available for exposing to the microorganism. Whereas for resistance, in the sense that they may not be able to metabolize the drug, with the result that the drug will accumulate in the body, drug will accumulate in the cells, so that will lead to further toxic effects. So it is important that it should be rationally designed, and it depends on the ability of these individuals to metabolize these drugs. And now we have the technology to identify those genes which produce enzymes which are involved in the metabolism of these drugs. This new area is called rational drug design based on pharmacogenomic approach, pharmacogenomics. Similarly, the gene therapy and control systems and the pharmacogenomic, where you produce is a custom-made drug. That is, you looking at the genome profile, genetic profile of an individual, the drug which is suitable for that individual has to be developed. It is more personalized medicine rather than a general medicine. Say, whoever goes to the physician with an infection, he will prescribe the same drug, the same dosage. It is possible to change from that generalized approach to a personalized approach, from a general medicine to personal medicine. And another thing is the earlier detection. That means from the earliest case, you go with the disease and then it is the treatment. Or to a certain extent, it is a preventive approach. It is a preventive approach. Now, from that preventive approach, you can go to a predictive approach. From a preventive medicine, you move on to predictive medicine. That is, if you know that people with such and such a genetic profile has the possibility of developing a disease, then that can be predicted earlier, with the result that appropriate, if possible, can be taken. So microbial genomics, I just indicated earlier. The risk assessment I have indicated. Then also there are other areas like bioarchaeology, anthropology, human evolution, and human migration, which can be studied from this genetic mix. This is just an example of the human migration. One of the earliest studies, in fact, almost at the time when the Human Genome Project was started, a classical experiment was done where they analyzed, they were looking for the evolution. They were looking for the evolution, where does it really origin, where does it, looking for the migration, where does it really start from. So the argument was that they started looking at instead of the genomic nuclear DNA, the nuclear DNA come from both mother and father.